Thanks, Isabel, and thanks everybody. We just saw in Jean's presentation, right? IRIS genebank is accessions are one of the most used. 13,000 is the number, right? So, but how to enable greater use across the table is something that wasn't clear until we started working on this. Is what we try to tackle with this tool. I'm gonna try to describe what we did, but that's essentially the reason why we did it. We wanted to enable greater use of collections by mining accession data. So those 13,000 accessions, they are delivered to whoever is requesting them, and those requests are coming often through email or through online systems. And what you see here in this slide is some examples of what those requests look like. I want banana accessions with high transpiration efficiency under osmotic stress. I want cowpea subset with drought tolerance. I want accessions for high elevations. And most of that information, drought tolerance, transpiration efficiency, that just doesn't exist in genebank databases, which are mostly, for the most part, they're limited to passport data. Yet, somehow the CGIAR is able to distribute 96,000 accessions to whoever is requesting, right? So there is, of course, a lot of expertise in the gene banks, right? And the collection curators, which is what is currently used to identify those specific subsets and address any of those requests. We wanted to build on that expertise and then build a tool that basically allows mining accession data to try to respond to some of these requests. This is how the CGIAR and a few additional national systems collections look like if you just plot them on a map. All of the accessions that have a geographic coordinate that's associated with them. That's how that map looks. Everything where there's green, there's been a collected accession. So the question is, how do you identify within this universe of accessions what is useful to you, right? And when you have a small group of accessions, it's very easy to separate. Imagine you have temperature and precipitation as two key indicators. If you have a very small number of accessions, then you can easily separate them by simple visualizations like we're seeing here. But the more accessions you bring into this analysis, and just to give an example, just the rice collection, and that's just one of the collections. Yes, it's one of the largest, but just the rice collection is 130,000 accessions, right? The combined data in Genesis, I think it's over 1 million accessions, right? So mining that cloud of data points is really challenging, can be really challenging. So what we did is we developed a method where you take the entire collection and then you create subsets by identifying specific characteristics. The method uses two steps, basically. The first step is you need to filter based on the passport data what you're interested in. So, for instance, I want all accessions from a particular taxon. That's a very simple thing to do. You can do it in Genesis. The second thing is by complementing the initial filter with mining. So using soil and climate data from publicly available sources, that data is extracted using the geographic coordinates, and then you can identify candidate accessions that could meet a particular criteria. Because there is an explicit evaluation data associated with the accessions, we can't be 100% sure, but the mining of the data allows you to identify for a number of adaptive traits what accessions could meet that criteria, right? So for example, I'm interested in maize accessions from Mexico that can grow at low temperatures. And we have a lot of evidence from peer-reviewed literature. Our colleagues from ICARDA who developed this tool together with us, they have been working on a methodology, developing a methodology and testing it. This methodology is called FIX, Focused Identification Germplasm Strategy. There's a lot of evidence that when you mine a collection in the way that we are suggesting here, you can identify a subset, and then that subset has a greater chance to give you what you're looking for, as opposed to a random subset, and especially so as opposed to a negative selection to the criteria you're looking for. How do we develop the tool? We wanted to understand the needs, right? So we wanted to talk to curators. We wanted to talk to other international gene banks outside of the CG and see how they did this. We talked to CGN in the Wageningen University. We talked to IPK. We talked to USDA. And we also reviewed a number of subsets which already existed in Genesis, which people had created, and then they just made available as specific data sets. We reviewed direct requests to gene banks. We also reviewed crop improvement program profiles to see what kind of adaptive traits people are looking for, are trying to bring into the breeding programs. And we also had direct interaction with gene bank staff from CGIAR, in particular CIAT and ICARDA, but also broadly with all of the CGIAR centers, because we did a survey to all of the collection curators and gene bank managers. So what we found in this interaction with the users is very simple, actually. There's a lot of nuance behind this, but it's very simple. And at the high level, it's high quality passport evaluation, especially for pest and disease, and characterization data is crucial to get the collections used. Without that, it's very hard that a collection will get used. And experiences from the CGN, IPK, and USDA, they show that the more interaction there is between what gets tested and what gets requested, what gets tested by the gene bank and what gets requested by crop improvement programs, for example, then the better use of the collection there will be. And generally, lack of reliable passport and phenotypic data will hinder the use of the tool and of the germplasm collections themselves. But there's also a lot of stuff that we asked and that we learned, right? One of these things is about the abiotic stresses that people find most important based on what types of materials they get requested. So heat and drought stress are two very important ones, especially because in many areas, agriculture is experiencing greater levels of heat and drought stress and combinations of those. But there's also soil-related stress, salinity tolerance, and things like that, photoperiod, and then also flooding. So with this, we then went back and tried to think, okay, what are the specific indicators that we're interested in, that we can bring, what data can we put together to try to help people identify subsets for these specific challenges. So this is when now the tool sort of comes to be developed. The subsetting tool basically allows you to build a custom subsets of accessions for multiple crops and environments using queries which are based on available data from Genesis, and while the tool itself is also available from Genesis. As a user, you can specify what specific stress you're interested in and what specific, with very little information, just what specific stress you're interested in and what indicators may be measuring best that stress, and then use that information just in the background, do a few analysis and statistics to try to then classify the collection into different groups according to those indicators. And then from those groups, you can then select what you're most interested in. One cautionary note here is we can only do this for accessions that have reliable coordinate data. We can't do it if we don't have that information. But there's a large part, in many collections, a large part of the collection has that information, fortunately. So what types of climatic and soil, agroclimatic and soil indicators we have? This is just an example here. I'm not gonna go through all of them due to time, but I invite everybody to go into Genesis and look at the subsetting tool. So as I said, drought, flooding stress, heat stress, photoperiod indicators, some soil indicators, and then there's also crop-specific indicators, right? That help tailoring some of the, for example, heat stress indicators to specific crops and species. And so this is an example for drought, right? For drought, what we have in the tool is 3 different indicators. You can select three of them. You can select just one of them, and you can combine these with others if your query is multidimensional, right? So it can get quite complex, and we allow for that flexibility. And the key is then the user gets to interact a little bit with the data, see what kinds of groups form, and then select from those, or just adjust their query if they wish. So this is just an example of the crop-specific indicators. I thought it was worth mentioning, right? So we have, for a few crops, just for the domesticated species, we have a couple of indicators, just counts of days for specific levels of temperature levels, high temperature, cold stress, low temperatures, and then what number of days is in the optimal range. So how does it do it, right? So basically you select the indicators and the initial filter. Imagine you're talking about wheat land races in a specific country, or in all of the Mediterranean countries, for instance. And then you select the indicators. Think you're looking at drought stress and heat stress, and you pick one indicator in each case. Then what it does is it clusters all of the accessions. It forms a series of groups, and then based on the characteristics of those groups, you can select them and just go for just then identify your subset. If the subset is too large, which it often is, if the size of the data set, if the size of the collection you're mining is too large, there's also an option where you can further subselect accessions into a more manageable set. Gene banks generally don't want to handle very big requests of hundreds and hundreds of accessions. So 25 to 50 accessions is mostly quite reasonable. So you can do that type of subselection with either randomly or manually, or using core collection method. So that's one way. The other way, this is currently not yet in the tool, but we're in the process of developing this. This feature will be ready by the end of this year, hopefully. And is you can also do it by similarity. We realize there's a lot of people going to gene banks. I am located in this site, and I want to grow this crop. Can you send me a few accessions that I can test? So we are developing a method that uses just the point location of where somebody is, and then can look at the climate similarity with all of the accessions that have been collected for a particular taxon of interest, or for a number of taxa of interest, and then help select based on the similarity what would fit best that environment. We can also do this using future climate data. What is the future climate of my site looking like in 2050s, and can you provide me accessions that could work under those conditions? That's also something that you can do, or vice versa. And this is just an example of how the tool looks like. This is an example using wheat landrace accessions. 50,000, around 58,000 almost landrace accessions, and there's what I selected here was drought stress, just based on total precipitation. And what you can see here, just for the entirety of the wheat land races, you get then the collection divided into 3 groups. You could also get it divided into more groups if you choose, if that's what you want. Or using this, you can then, if you're happy with either one of these groups, then you can select it, and then look at the specific accessions which are there. And in Genesis, there's an additional feature, not part of this tool, but just part of the Genesis system, where you can select and then put them in your shopping cart and order them. And then those queries go to Genesis, and then they get transferred to the specific gene bank that is holding those accessions. So that's it from me. Thanks.